Good morning, and thank you for joining me for this webinar on optimizing soluble protein expression using codon optimization, RBS design, and expression vector design. By way of introduction, my name is Rachel Spear, and I'm a technical writer here at GenScript. I focus on our gene synthesis and molecular biology services. I did my PhD work in the neuroscience program at Weill Cornell, where I did a fair amount of molecular biology to create DNA constructs for stable or transient expression of recombinant fusion proteins in mammalian cell lines for use in reporter assays and biochemical analysis. I also worked alongside colleagues who needed to purify large quantities of enzymatically active protein from an E. coli system for in vitro enzyme kinetic studies. So as you might imagine, we ran into our fair share of concerns about getting the right DNA constructs and getting sufficient protein expression. And I confess that as a graduate student, I was completely unfamiliar with codon optimization, gene synthesis, ribosome binding site design. I never thought about optimizing my expression vectors. I just cloned new cargo into the same vectors that the lab had always used. So I'm really excited that you all are attending this webinar so you can learn more about codon optimization and RBS design and how optimizing your expression vectors really can be worthwhile to help your research. So if you have a question at any time during this webinar, please type it into the chat function. After the presentation, I will read through the questions that have been submitted and do my best to answer them. This webinar, including the Q&A, will be archived on our website. Okay, so my goal today is to introduce you to 3 easy ways to improve your yield of soluble protein. The first is codon optimization, which can be used in any expression host system. And I'll spend the most time on this because I'm really excited to let you know about some updates that we've made recently to our algorithm for E. coli expression. The second tool I'll talk about, ribosome binding site design, is only going to be useful for those of you who are doing prokaryotic expression systems, such as E. coli. The third topic, which again applies to any expression host, is optimizing your expression vector. After I introduce the principles of these 3 tools, I'll point you to some additional resources for further learning, and I'll describe some services that GenScript offers that can help you get better protein expression in your research projects. So let's start with some context. This is an overview of your workflow when you're trying to express a protein. First, you select or design the amino acid sequence you want to express, and you decide on an expression host. Then you design the DNA construct that will serve as your expression clone. Then you get it into your cells, express your protein, purify it, and finally characterize it. Each of these steps is important in determining the final protein yield. And at the end of this presentation, we'll return to this workflow so I can point you to some other resources for optimizing each of these steps. But for today, we're going to focus on optimizing the expression clone. All 3 of the tools I'll tell you act at this step in your workflow. Within your expression clone, there are many different factors that you can choose to tinker with. For example, you may want to change or shorten your amino acid sequence. For example, researchers often use truncated versions of proteins if the ultimate goal is getting a crystal structure of a specific domain. Mutations or truncations would be considered protein design, and that's outside the scope of today's discussion. Okay, what we will talk about, for example, is that you may want to optimize the promoter that you use to drive expression. We'll talk about that in part 2 and part 3. You may also want to try different affinity tags or solubilization tags, which are often incorporated into expression vectors 5' or 3' to the MCS where you clone in your target gene. Again, we'll talk about that in part 3 along with selecting the best vector and optimizing your vector. Our first topic today is going to be codon optimization. We can define this as introducing synonymous mutations that favor efficient soluble protein expression. Codon optimization is different from protein design because codon optimization only changes the nucleotide sequence, but not the amino acid sequence. Codon optimization can be performed across the entire nucleotide sequence that will be translated into protein. So that can include your tags as well as your target protein. So first, why do codons matter? Well, as you know, there is redundancy in the genetic code with 64 possible trinucleotide codons that correspond to only 20 amino acids plus the stop codon. This means that you can introduce changes in the nucleotide sequence, most often in the third nucleotide of a codon, that will not change the amino acid. These are called synonymous mutations. And while synonymous mutations do not change the amino acid sequence, they do change protein expression levels. This fact has been observed many times in virtually every species. In fact, synonymous mutations can also alter protein conformation, post-translational modifications, protein stability, and consequently protein function, and thus the cell's behavior and survival. So when we design a DNA construct from which we will express a protein, we can choose to introduce synonymous mutations that will favor efficient, soluble, functional protein expression. That's our definition of codon optimization. Now, numerous studies from the last decade have examined how exactly synonymous mutations affect protein expression, and there's now a great deal of evidence that the nucleotide sequence influences the ultimate yield of soluble protein through diverse mechanisms at different stages of the process, from transcription to mRNA processing and stability to translation initiation and elongation, and even protein folding. 5 or 10 years ago, many people were focused on the goal of adjusting expression constructs to mimic the codon bias of the host. For example, by replacing the rare codons with preferred ones. I think the biggest surprise has been that codon usage bias is not the only factor. You can see it listed here among many other factors. In fact, in many cases, codon usage bias is not even the most important factor for determining protein yield. We also need to design DNA sequences to not contain certain sequences that have been revealed to cause problems, such as SD-like sequences or AT-rich sequences that can cause premature transcriptional termination. Or there's a specific 7-nucleotide sequence that's known to introduce frameshift mutations. So codon optimization is much more than just adjusting for codon bias. GenScript's codon optimization algorithm is designed to take into account all of these evidence-backed factors, as well as to offer flexibility to adjust the weight of different factors, or to add customized constraints, such as filtering out restriction sites that you're planning to use in your cloning strategy, or reducing similarity between different sequences if you're creating a library. In some cases, you may want to specify an alternative codon table if you think your experiment can benefit from that. For example, there have been publications reporting that for certain amino acids, researchers have empirically observed a preference for a codon that's not the most abundant. So some of our customers who order gene synthesis regularly will actually develop a standing protocol for codon optimization according to their particular needs. So we can definitely accommodate your requirements. Okay, so how do we do this? Well, we actually hold a patent on our method for using a particle swarm optimization algorithm to power our optimum gene technology. We do an in silico evaluation of over 10,000 candidate sequences in each round of analysis, and we use a complex sorting matrix to select the best sequence for each gene optimization task. So we've actually done extensive wet lab validation of our algorithm as well. And we find that codon optimization improves expression in all host systems, including E. coli, yeast, insect baculovirus, and mammalian cells. Here's some of our R&D data using the particle swarm optimization to improve production of single domain proteins. This is showing 4 native gene sequences which code for 4 different proteins that were difficult to express. We optimized them using Optimum Gene. And after codon optimization, you can see from these gels that we get vast improvements in expression compared to the native sequences. We also find that codon optimization is beneficial for more difficult projects where we're trying to produce protein complexes. Here we're showing data for a recombinant protein with 3 subunits, which is technically very difficult to express. We optimize the sequence for 3 different hosts, E. coli, insect, and mammalian. This is using human cells. And we obtained high expression levels in all 3 expression systems using, again, Optimum Gene. We also often get requests to express protein complexes for crystallography studies. So here's an example of production of the ligand binding domain of a nuclear receptor associated with its ligand in the correct conformation, again, after using codon optimization to boost expression in the E. coli host. A final example I want to show is that codon optimization improves monoclonal antibody production in a mammalian expression system. Here you can see on the left, in a transient expression system, we tested 3 different recombinant antibody proteins. You can see that after codon optimization, the expression level was increased between 2-fold and up to 30-fold. In a stable expression system on the right, our codon optimization technology significantly improved the yield of our pool of positive clones. You can see from how this curve is shifted that a much higher share of the clones were producing significant levels of MAB. And when you want to pick a few clones to expand for scale-up, you definitely want to be choosing from these clones that are producing around 4 grams per liter compared to the native sequences, which max out about 2 grams per liter. So GenScript has actually used our optimum gene algorithm to optimize over 500,000 sequences in all major expression systems. You can see that most of our work has been in mammalian and E. coli, but all of them we're quite experienced in. We routinely test and improve our algorithm, both because we want to provide the best results for our customers, and also because we use our codon optimization algorithm in-house for our protein services. Our protein department has delivered over 5,000 proteins in 4 expression systems, and our track record shows a 95% success rate for all protein projects. And this includes projects on a wide variety of types of proteins shown here, including difficult to express membrane proteins, as well as proteins of all different classes. So now we're really excited to announce the launch of a new update to our optimum gene algorithm for E. coli. This version is called Apricot, and we rolled it out in May, and we've gotten great feedback from our customers about it so far. The differences between our older Opti10 algorithm and this year's new version are twofold. First, instead of using the standard E. coli genome codon table, we use our proprietary high productivity codon table. And in addition to performing global codon optimization across the entire CDS, we adjusted the threshold for accommodating N-terminal codon bias according to some recent research findings. So let me show you the results of our wet lab validation with this new algorithm in E. coli. So you can see here across a test sample of 16 highly varied proteins, the new algorithm gave the same or better yield compared to the old algorithm in all but one case. Half of the proteins we tested saw significant increases with the new algorithm, in some cases on the order of 50% improvement, but in other cases we saw 5-fold or more improvement. And again, this is compared to our previous optimization algorithm, which is already a big improvement compared to the native unoptimized sequence. Of course, we're not the only company to offer codon optimization, so we routinely compare our sequences to those generated by other codon optimization algorithms. And our in-house data suggests to us that Optimum Gene really is more effective at increasing protein yields than our competitors' methods are in half of all cases where we tested. And there's only one protein that we've tested where our version showed lower expression than a competitor's optimized version. So I think the numbers kind of speak for themselves. Again, given how powerful codon optimization is and how GenScript really has the best expertise in this, many researchers don't even bother testing the wild type sequence first or comparing different codon optimization algorithms. They just start by ordering a codon optimized gene from GenScript to save time. Hundreds of papers cite GenScript for codon-optimized gene synthesis, and you can see these papers span a variety of journals and research fields, as well as a variety of particular reasons for pursuing codon optimization. You can find many more examples by searching the citations database on our website, which contains, I think, over 12,000 papers now that cite GenScript services and products. So now that we've talked about codon optimization, which again, I'll be happy to answer questions about at the end if you have them, and I can also point you to some more resources to learn more. I realize we went through that pretty quickly, but let's go ahead and move on to the next design tool we're going to talk about, which is ribosome binding site design. And again, this is only relevant to prokaryotic expression systems like E. coli. So I want to start with a review of protein expression in a nutshell. As you know, the rate of protein production is determined by the rate of transcription of DNA to RNA and then translation of the RNA into protein. However, protein production in a cell has to be balanced with other cellular processes, such as the rate of protein folding and the rate of secretion or insertion into a membrane. And so sometimes you want to increase your rate of protein production, but sometimes you might actually need to decrease it in order to balance it against those other cellular processes. So when you think about modulating the rate of protein production, you have to understand what the rate limiting step of protein production is. And it turns out that it's translation initiation. Translation initiation is mediated by the binding of the 30S ribosome to the area of the mRNA that spans between the 5' upstream region and the start of the CDS region. So you can see in this diagram that there's binding between the Shine-Dalgarno sequence and the ribosomal complex itself, but the ribosome really has a much larger footprint than just the SD sequence. So when you want to maximize your protein titer, again, it's a balancing act. If you think, well, we want to improve, let's just say that you're starting by observing that your expression level is too low. You're getting low protein titers. That may be because your translation initiation rate is too low. However, if you try to improve that by just increasing your translation initiation rate, you may get very high levels of translation, but that may clog the machinery for folding the protein properly. So you may end up with a lot of improperly folded protein, and again, not really seeing an increase in your titers of properly folded soluble protein. So what you really want is sort of the just right optimal peak between expression level and properly folded protein. So again, in some cases, you may want to increase your protein production level by increasing your translation initiation rate. But in other cases, you may actually want to slow down translation for better solubility of E. coli expressed proteins. An RBS design can let you do either. So when you look at factors that affect translation initiation, a lot of research has been done on this, and it turns out actually that there are many different factors. And these factors depend not only on the 6 or 8 base pair Shine-Dalgarno sequence, but also upon sort of a 35 nucleotide region upstream of the start codon, and also the first segment of the CDS downstream of the start codon. And there are many different molecular interactions that are happening and that can influence the rate of translation initiation. Some of them are listed here. Basically, you've got to think not only about hybridization between the mRNA and the ribosome, but also any features of the mRNA that might block the ribosome from binding, or the distance between certain features on the mRNA that might cause the ribosome to have to stretch or compress. And so this is really a complex problem to solve. So for example, let's say that you've got a sequence that you're starting with and you'd like to increase the translation initiation rate. So you say, well, actually, this RBS region doesn't have a consensus Shine-Dalgarno sequence in it. So maybe if we add one, that will maximize the interaction between the mRNA and the ribosome, and we'll be able to boost our expression level. Well, in this case, you can see, when you added the Shine-Dalgarno sequence, you actually created a new mRNA structure of a hairpin loop that will inhibit translation initiation. So you can see that it's not as simple as just sort of looking at it and being able to do the design by eye, because there are many overlapping causes which make it difficult to design this by eye, basically. Instead, you need to use a quantitative model that will calculate all of the causes and predict the best outcome. So Howard Salas at Penn State has worked very hard on this to create a biophysical model of translation initiation for bacterial microRNAs, or messenger RNAs, I'm sorry. And that model will take as inputs your RBS sequence and your CDS sequence, and it will output an RBS that has been designed for you to give you translation initiation of the rate that you desire. first, I'll walk through the steps for how it does that. Okay, so we use this automated design to optimize our expression. So here's an example. Let's say your objective is to design an RBS sequence that will decrease translation initiation of a specific protein 20-fold. So here's your initial sequence. And if you want to manipulate that 35 nucleotide 5' UTR region, there would be a very large number of possible variants that you would have to try. So obviously you don't want to spend the time screening 4 to the 35th variants. Instead, you can use computational optimization to find 1 or a small number, maybe 5 variants that you would like to test in the wet lab. So the way that the computational optimization works is that you start with a target protein production rate that you want to achieve and then you translate that into a target translation initiation rate, given the fact that translation initiation is the rate limiting step. Then you can have this computer program do in silico mutations and then evaluate the predicted translation initiation rate for each sequence and accept or reject it, and then do iterative rounds of this kind of computation to finally return a designed RBS sequence for your needs. So let's look at an example of this. If you want to reduce your translation initiation rate 20-fold, and you put in your initial sequence, let's say that it gives you, that the calculator predicts that with your original sequence, your translation initiation rate would be in arbitrary units 400,000. So if you want to decrease that rate 20-fold, then you want to design a new RBS that would have a predicted translation initiation rate of 400,000 divided by 20 is 20,000 arbitrary units. So the RBS calculator can give you a sequence that meets those criteria. On the other hand, if you don't know what your target translation initiation rate ought to be, you don't know if you ought to increase or decrease, or you know you need to decrease it, but you don't know by how much, then you could put in several different target TIRs, like in this case it's shown 100, 300, 3,000, 10,000 up to 100,000, and you could get this nice small set of 6 variants that span a nice range to allow you to do very small scale, not very labor intensive, quick screening to find out exactly what part of that region you want to be in. So again, the ribosome binding site calculator takes your RBS sequence and your CDS sequence as inputs, puts it into a biophysical model of how translation initiation works, and predicts the translation initiation rate. So how does that prediction hold up in the wet lab? Well, on the left, you can see the wet lab validation that was performed by the creators of the RBS calculator, Howard Salas and his lab at Penn State. And they used a very rigorous set of, I think, 624 different proteins with many, many, many different parameters. They really sort of threw the kitchen sink at this to try to test it under the harshest conditions. And you can see that they got a very nice linear relationship between predicted translation initiation rate and measured expression levels. It's a little bit messy, but the R-squared value is 0.76, so pretty good. When other people, third parties, have done their own validation, they actually see much tighter correlations between the calculated RBS strength and the measured rate of translation. And there are some references there if you'd like to learn more. Again, the RBS calculator is only for use in prokaryotes, but it has been tested and designed to work across many different popular prokaryotic species. Different organisms have slightly different details of their preferred ribosomal binding sites, but basically the same RNA biophysics. So here you can see the data using the RBS calculator in E. coli BL21 cells, and then in 3 other types of cells that are commonly used, especially for industrial biotechnology. So how exactly does the RBS calculator work? Again, it takes your mRNA sequence input and puts it into a thermodynamic model that actually calculates the Gibbs free energy of the interactions, not only between the ribosome and the mRNA, but many other molecular interactions that are known to be important. And then it outputs your translation initiation rate on a proportional scale of arbitrary units. In this diagram, I just want to point out again that the ribosomal footprint spans the RBS sequence in the 5' UTR and also the beginning of the CDS sequence. So RBS, ribosome binding sites, are not modular units like promoters that can be swapped in and out without any harm, where you can have sort of a strongly acting one and a weakly acting one and swap them out. The ribosome binding site has to be custom designed for every CDS that you're going to use. So fortunately, the RBS calculator makes it quite easy to do this. And GenScript has actually partnered with Howard Salas's lab to offer RBS design services. Again, keep in mind that the RBS is sequence dependent. You have to design the RBS after you've decided on what your CDS is going to be, which means if you're going to do codon optimization to your CDS, you need to do the codon optimization first and then design the RBS for the codon optimized version. RBS design can be used for single proteins and also for multi-enzyme pathway optimization. And our technical account managers can definitely help you with your particular needs. Here's just sort of a quick guide if you don't even know where to start, depending on what kind of protein you're working with and what sort of yield you're hoping to achieve. Here's a little quick guide to help you know what sort of range of arbitrary units you might want to target for your first RBS design project. So that's been a really quick run through of RBS design. I want to move on to our last topic, which is expression vector optimization. Again, most people don't even think about changing the expression vector that they're used to using. And when they look at their plasmid maps, all they really focus on is making sure that the sequence of the target is correct. But I want to encourage you to go back to your vector maps and look at all of the components and think about whether swapping expression vectors or customizing the vector you're using to swap out one of these components might actually be worth your time and trouble. So just a quick review of what's in an expression vector. Expression vectors have to achieve many different goals, and they contain many components to help achieve each of those goals. So first of all, you need to be able to insert your cargo into the plasmid vector and verify the insert sequence accuracy. So you can do that using the multiple cloning region, or multiple cloning site, MCS, of the expression vector. Most people are still using restriction sites with restriction enzymes because they're extremely easy and flexible. You also want to make sure that your vector includes the 5' and 3' primer sites for doing sequence verification. For the next goal in your experiments, for inserting the plasmid into cells, enabling the plasmid to replicate inside the host, and selecting the cells for carrying the plasmid, there are a number of components that are important for that. For transcribing the mRNA from the plasmid, doing your DNA to mRNA transcription, you've got to have a promoter to activate transcription. You could use a constitutive promoter, or you might want to use an inducible promoter. You've also got to think about the terminator at the 3' end of your target sequence, because transcription termination is really important for making sure that you get full-length sequences that are transcribed and released at an efficient rate. Then when you translate the mRNA into protein, that's when you're going to look at the ribosome binding site. We talked about the Shine-Delgarno sequence and the surrounding regions in prokaryotes. In eukaryotes, most people use a consensus Kozak sequence, but you can swap that out. And then in order to promote proper folding of the nascent protein, you have a couple of different options for how you could do this. You might want to try co-expressing chaperones. You might try using solubilization tags, or you might try using codon optimization or other types of modifications to improve proper folding. And finally, when you need to detect or purify your target protein, you want to look for epitope tags or reporters, which again will often be incorporated into your expression vector. So just a quick review, there are many different transcriptional promoters that can be used. And this transcriptional promoters are often sort of modular parts that you can easily swap in and out. These are just a few of the commonly used promoters, but you might want to look at what promoter you're using in your expression vector and think about trying a different one. Epitope tags or fusion proteins are also commonly used to help with detecting target protein, with purifying target protein, or with improving the solubility of target protein. If you're not using one of these, you might want to consider using it. If you're using one and you're having trouble with it, then you might try a different one. So if you've got a His6 affinity tag, but you're having trouble with purifying, you might try a different tag and a different purification scheme. And if you are worried about how the tag might be affecting the folding or the enzymatic activity of your final protein, you might consider using tags that can be removed at cleavage sites that are placed between the target and the tag. So in the interest of time, I'm going to point you to learn more about expression vectors from a webinar that I presented a few months ago. If you are relatively new to molecular biology, if you're a grad student in the lab and you're just starting to design expression clones for the first time, this could be really helpful for walking you through how to read a plasmid map and giving a lot more detail on those components that we just ran through very quickly. The ones that I selected to run through here are the ones that are most often optimized either by swapping to a new expression vector or making changes to the vector that you're using. So that's been a bit of a tour de force. I just want to back up a little bit and summarize how to optimize protein expression. A big key that I want to remind you is that optimizing is not the same thing as maximizing. More is not always better, especially as we saw in the middle of this presentation when we talked about soluble protein with RBS design. So some steps that you might want to consider are optimize the expression vector, which could involve reorganizing components within the vector or trying a new vector altogether. Keep in mind that if you're using prokaryotic expression vectors and you want to optimize the RBS, it must be customized for each new cargo that you're going to test. Of course, you can optimize the cargo, as we talked about at first, with codon optimization. And then some other things that you might consider optimizing that we haven't discussed today are you might try optimizing the host. Sometimes switching the platform that you're using or just switching the strain of E. coli that you're using can be really helpful. So you might recall that I showed our in-house protein scientists often try expressing for protein complexes or for difficult proteins, they'll often try expressing them in 3 different systems, in E. coli, mammalian, and insect, and then see which one works best and go forward with that. And then, of course, you can optimize your methods for transformation, selection, growth, induction, purification. There are many different variables that are in your hands. So I just want to summarize that GenScript offers a pretty comprehensive toolkit to help you optimize your expression. We offer codon optimization as a free service with our gene synthesis. As we discussed, codon optimization can help boost both transcription and translation in any host system. We're pleased to offer RBS design using the RBS calculator to help you tune translation initiation rate in prokaryotes. And finally, to help make it easier for you to test different expression vectors, we offer express cloning that lets you choose from over 150 free vectors to try. So it's pretty low risk way to try multiple vectors to find the one that works best in your system. I also want to point you to some related webinars. If you want to learn more about RBS design, the inventor of the RBS calculator, Professor Howard Salas, gave a webinar for us just a few months ago, and that's archived on our website. Last year, I gave a webinar on codon optimization, going into much more detail and showing a lot of case studies, which I didn't have time for today. So if you'd like to see more examples of that, check that out. My colleagues, Kashev and Leanne, have presented some really wonderful webinars for how to optimize the conditions for recombinant soluble protein expression, especially focusing on E. coli hosts, but really across multiple host systems. And finally, another step that we didn't talk about today is there are strategies to help promote proper protein folding. For example, it can be improved by chaperone co-expression. One of our senior scientists gave a great webinar about that a few months ago. As you know, folding is coupled with translation initiation and elongation. And so the tools that I talked about today, RBS design, can help with regulating the translation initiation, and codon optimization can help modulate the rate of elongation. You can either try to optimize codons to speed elongation or de-optimize them to slow down the rate of translation and perhaps allow elongation to happen and prevent the formation of truncated peptides, polypeptides. Okay. So how do we actually do all these optimizations, codon optimization, RBS optimization? It's really powered by gene synthesis. Instead of having to go in and do site-directed mutagenesis every time you want to optimize a nucleotide, it's much easier and faster to simply do de novo gene synthesis to create the custom insert that you want. GenScript is a leader in gene synthesis. We offer multiple flexible services depending on your price and turnaround time needs. As I mentioned, we also offer cloning of your synthetic gene into any vector. We offer 151 free expression vectors for you to use. The cloning service is just $49 and takes 2 days, and you can pick from any of these vectors, or you can try out multiple vectors. In addition to express cloning with the 151 popular expression vectors, we also offer vector art cloning if you have a custom vector that you've created in your lab and you want us to store your custom vector and use that for cloning your genes going forward. We also offer express mutagenesis services if you just need to make a simple change. So for example, swapping in and out ribosome binding sites is often done using mutagenesis because it's just a 35 nucleotide region. And so that's easily done with our mutagenesis service. And then finally, if you're doing genome editing, we offer a number of CRISPR-related services as well. So again, to sort of bring this together, today we mostly talked about how to design and manufacture your expression clone, that is the plasmid DNA construct that you're going to use. But for all of the downstream steps, GenScript also offers full service solutions, including our potential protein expression evaluation system to let you decide which host system you want to use based on doing small scale expression in different hosts and comparing the results. We also can do the protein expression for you in your choice of expression host system. We can also help with getting purified protein in difficult cases by using our FoldArt refolding technology or our toxin eraser endotoxin removal services. And we also have protein characterization services. So I want to thank you again for your attention and remind you that GenScript is very proud to be the most cited biology CRO offering services across the whole spectrum of life science reagents, as well as some catalog products and discovery biology services as well. And at this point, I would, again, thank you for your attention. I would ask you to please complete the survey that will pop up on your screen when you close the webinar. If you have any specific questions or feedback on this webinar, please feel free to email me. This webinar will be archived on our website, and all of the other archived webinars that I mentioned, you can also find them there. And at this point, I will take questions from the audience. So let me see what we have here. Okay, you mentioned codon usage bias. Can you explain what that is? Oh, that's a great question. Yeah, I'm sorry I gave kind of short shrift to that today. So to answer the question, in every species examined to date, all of the 64 codons are not represented equally throughout the coding regions of the genome. There's a bias towards certain codons and not others to be used to encode any given amino acid. There's a corresponding bias in the abundance of tRNA isoacceptors. So the tRNAs for the more commonly used codons are themselves more abundant. And this bias is species specific. So, for example, it was noticed that when you try to express a protein from one species in a different host, for example, if you try to express a human protein in E. coli, as we often do, heterologous protein expression levels are often very low. And one explanation for this could be that you have a mismatch in the codon bias of the human gene versus the E. coli host. So the presence of codons that are rare in E. coli could reduce protein expression. And the mechanism for that could simply be the relative rarity of those tRNA molecules, which could cause translation to stall. So there may be long pauses during the elongation phase that could really hurt you. So that's the theory of codon bias, is that different species have different codon biases. And so codon optimization initially focused on trying to replace rare codons with preferred codons in order to sort of overcome that codon bias. But as I mentioned, now we know that codon bias is not the only factor, and in fact, it's often not even the most important factor in determining how well a sequence will be expressed. So thank you for that question. And I would, if you want a lot more detail about the rich and sometimes contentious history of codon bias and codon optimization, I would really refer you to that archived webinar. So let's see. Are there different codon optimization algorithms for different species of mammals? For example, is difference between CHO, which is Chinese hamster, and HEC, which is human, for antibody expression? So absolutely, yes. So our codon optimization algorithm uses the reference genome of each specific host species, and there are publicly available codon tables that will show the codon bias in each species genome, and we have those codon tables freely available on our website for anybody to use. We also have our proprietary codon tables, which basically are adjusted to account not only for how prevalent are these codons in this genome, but how productive are they? How much they influence translation? Again, this gets back to the question of codon bias, where just because a codon is commonly used doesn't necessarily mean that it's the one that makes translation proceed in the speediest or most efficient way. So there are publicly available codon tables. We have our proprietary codon tables that inform our algorithm. But yes, absolutely, we use different reference tables for different species of mammals according to the specific genome, definitely. Okay, so then, oh, I see, I'm sorry, there was a question that was asked sort of halfway through that says, will the entire presentation be on expression in E. coli? I'm sorry that the RBS really was specific to E. coli, but codon optimization, section 1, and expression vector optimization, section 3, those really are relevant to mammalian or any other host system. So I hope that you found this useful, even if you're not working in E. coli. And if you have specific questions about non-bacterial systems that you want to ask, I'm absolutely happy to take those. Okay, somebody asked, I have ordered some codon optimized sequences. Can I order RBS design and attach it to my previously ordered sequences? Or is it possible for me to design RBS using your online RBS calculator and do cloning myself? So great questions. Yes, you can definitely order RBS design for your previously ordered sequences. Um, Again, the RBS region is about 35 nucleotides in the 5' UTR, and so we can take your previously ordered clones and just do a simple mutagenesis to swap out the RBS region. Um, in terms of you doing it yourself, you absolutely could do that. Unfortunately, we don't have the whole online RBS calculator available on our website. It's a little bit complicated, and so we have our technical account managers actually use the calculator to give you your output. So if you have specific constraints that you'd like a little more flexibility to do it yourself, absolutely communicate those to your normal GenScript rep, and they can help you get exactly what you need. But in terms of ordering RBS design for previously synthesized sequences, that's really easy to just do a simple mutagenesis to swap those out. Let's see. Why would changing the DNA sequence alter post-translational modifications, protein stability, folding, etc.? That's a great question. I, again, would refer you to the archived webinar on codon optimization. I'd also be happy to email you some specific references to check that out. But the basic answer is, protein folding is in part determined by the rate of translation and elongation, because there have to be pauses in translation elongation to allow time for folding to occur properly. And so sometimes you get misfolding because translation is running too fast. And then in terms of protein stability, again, that can be determined by folding. And post-translational modifications, again, can be determined by the precision of folding. And again, cell viability can be determined by, PTMs, which are determined by stability and folding. So there's actually, I saw some great talks at the biophysics conference last year about how the folding actually occurs, and how sort of at very precise levels the rate matters for when new residues are available, sort of in the translational and folding complex. So I'd be happy to discuss that in more detail if you'd like. So I think there's one more question I see here asking about Arctic Express strains. I'm going to follow up with you individually on that one. And then I think that's all the questions that I see from the audience. So I want to thank you so much for your time and encourage you again to take that survey, to send us feedback by email. And we wish you the best with your research going forward. Have a wonderful morning.